Characteristics features of RNA polymerase of phages. RNA polymerase of phages are enzymes responsible for the synthesis of RNA from DNA templates during the replication and transcription of phage genetic material. These enzymes exhibit several characteristic features that distinguish them from other RNA polymerases found in cellular organisms. In this essay, we will explore the significant characteristics of RNA polymerase in phages and their implications for phage biology. The structure of RNA polymerase of phages are typically multi-subunit enzymes composed of several protein subunits. The exact composition and arrangement of subunits can vary among different phages, but they generally consist of a catalytic subunit and various auxiliary subunits. The catalytic subunit is responsible for the actual synthesis of RNA, while the auxiliary subunits assist in promoter recognition, processivity, and other regulatory functions. Diversity of RNA polymerase. Phages are a diverse group of viruses infecting various hosts across different environments. Correspondingly, their DNA polymerases show significant variation in their sequence, structure, and function. Phages can be broadly classified into three groups based on the types of RNA polymerase they encode: single subunit RNA polymerase, multi-subunit RNA polymerase, and RNA-dependent RNA polymerase. Single subunit RNA polymerase, meaning phages, especially those infecting bacteriophages, encode single subunit RNA polymerases. These enzymes are relatively small in size and are responsible for both replication and transcription in phage genes. Multi-subunit RNA polymerase, some phages like the T4 bacteriophage encode a multi-subunit RNA polymerase. This polymerase consists of several subunits that work together, enabling more complex regulation and fidelity during transcription. Multi-subunit RNA polymerase are often found in large and more complex phages. Then RNA-dependent RNA polymerase. Many RNA phages and some DNA phages encode RDRPs, which play a crucial role in the replication of their RNA genomes. RDRPs catalyze the synthesis of RNA strands based on an RNA template, a unique feature not found in cellular RNA polymerase. Then promoter recognition. Phage RNA polymerase recognize specific DNA sequences called promoters, which initiate transcription. These promoters are typically located upstream of the genes and to be transcribed and contain conserved nucleotide motifs recognized by the polymerase. The auxiliary subunits of the RNA polymerase play a critical role in promoter recognition by interacting with the DNA sequence and facilitating the binding of the catalytic subunit. Phage RNA polymerase have evolved to recognize specific promoter sequences on the phage genome. These promoter regions typically contain conserved elements that are recognized by the polymerase initiating the transcription process. The specificity of promoter recognition is vital for the temporal and spatial control of genes expression during the phage life cycle. Then rapid initiation and elongation. Once the RNA polymerase binds to the promoter region, it initiates transcription by melting a DNA double helix and forming a transcription bubble. The catalytic subunit of the polymerase adds nucleotides to the growing RNA chain following the template strand of the DNA. During initiation, the RNA polymerase undergoes conformational changes that allow it to transition from a closed complex where the DNA is intact to an open complex where the DNA strands are separated. Phage RNA polymerases are known for their rapid initiation and elongation rates. These characteristics allow phages to efficiently produce a large number of RNA transcripts during infection, promoting the takeover of the host cell's machinery and ensuring a high burst size of progeny phages. RNA polymerase of phages exhibit high processivity, meaning they can efficiently synthesize long RNA molecules without dissociating from the DNA template. The processivity is facilitated by the interaction between the catalytic subunit and the auxiliary subunits, as well as the stability of the transcription elongation complex. Some phages RNA polymerase also possess intrinsic proofreading activity, allowing them to correct errors during RNA synthesis. Phage-specific transcription factors. Some phages encode their own transcription factors that interact with the RNA polymerase and modulates its activity. These factors can enhance or repress transcription of specific phage genes, allowing for precise control of gene expression throughout the infection cycle. Then termination of transcription. Phage RNA polymerase employ different mechanisms of transcription termination. One common mechanism is the formation of a hairpin structure in the newly synthesized RNA molecule, followed by dissociation of the RNA polymerase from the RNA template. Another mechanism involves the binding of termination factors that cause the release of the RNA polymerase from the DNA template. The choice of termination mechanism can vary between different phages and often influenced by specific regulatory signals within the phage genome. Error-prone replication and mutation rates. Phages are subjected to higher mutation rates during replication, mainly due to the lack of proofreading mechanisms in their RDRPs or the use of error-prone DNA polymerase during replication of their genomes. This high mutation rate contributes to the evolution and adaptation of phages to their dynamic environment, leading to the emergence of new phage variants in altered host specificities and virulence factors. Then template switching. In the case of RNA phages that utilize RDRPs, template switching is a unique feature during replication. The RDRPs can switch templates during RNA synthesis, resulting in the formation of chimeric genomes containing genetic information from different phage variants. This process, known as recombination, contributes to the generation of genetic diversity and the emergence of new phage variants with different properties. And antibiotic and antiviral targets. Because of the difference between the phage and the host RNA polymerase, these viral enzymes present potential targets for the development of antiviral agents. But selectively inhibiting phage RNA polymerase, it may be possible to disrupt the replication and transcription process, leading to the inhibition of phage growth and the prevention or treatment of phage infections. Regulation of RNA polymerase activity. The activity of phage RNA polymerase is tightly regulated to ensure proper timing and control of gene expression. Phages have evolved various regulatory mechanisms to modulate the activity of RNA polymerase. For example, some phages encode transcriptional repressors that bind to specific RNA sequences and prevent RNA polymerase binding to promoters. Other phages utilize regulatory proteins that interact with RNA polymerase subunits and modulate their function. Additionally, the availability of nucleotide triphosphate and other cofactors influence the activity of RNA polymerase. In conclusion, RNA polymerase of phages possess characteristic features that enable them to efficiently transcribe the viral genome and regulate gene expression. Their multi-subunit structure, promote recognition ability, processability, and termination mechanisms all contribute to their essential role in phage replication and protein synthesis. The understanding of these characteristic features provide valuable insights into the molecular mechanisms of phage gene expression and can contribute to the development of antiviral strategies targeting phage RNA polymerases. Then processability. Processability refers to the ability of an RNA polymerase to remain associated with the DNA template during elongation and synthesizing long RNA molecules without dissociation. Phage RNA polymerase often exhibit high processability, allowing them to transcribe entire phage genome efficiently. This property is crucial for phage replication and the production of viral RNA. Then evolutionary relationships. Comparative studies of RNA polymerase from different phages have provided insights into their evolutionary relationships and the relatedness of phage groups. By examining the conserved and divergent groups of regions of phage RNPs, researchers can trace the evolutionary history of phages and their interactions with bacterial hosts. This knowledge contributes to our understanding of phage biology and their potential applications in biotechnology and medicine. Then minimalist structure. Phage RNA polymerases are relatively smaller and simpler in structure compared to the RNA polymerases found in eukaryotes or bacteria. They usually lack some of the subunits present in their cellular counterparts, which allows for efficient transcription in the confined phage capsid. This minimalist structure enables the phage to package the RNA polymerase within its viral particle efficiently. Then pausing and anti-termination. Phage RNA polymerase has evolved specific mechanisms to regulate transcriptional pausing and anti-termination. Pausing allows for precise regulation of gene expression, while anti-termination is employed to overcome transcription termination signals, enabling the synthesis of longer transcripts. These regulatory mechanisms are essential for the controlled expression of phage genes during the different stages of the viral life cycle. Then phage-specific modifications. Phage RNA polymerase often undergo post-translational modifications that confer unique properties. And such modification is the addition of unusual nucleotides such as hydroxymethyluridine or glycosylated bases to the RNA product. These modifications can influence RNA stability, translation efficiency, or protect the RNA from host immune response. Phage RNA polymerase may also be modified with accessory proteins that further enhance their functionality.